Hola, ¿qué tal? Muy buenas tardes a todos. Bienvenidos a una reunión más con nuestro programa Come, Listen and Learn, que trabajamos con el Departamento de Inglés de la Facultad de Medicina Veterinaria y Zootecnia. Voy a dejar los micrófonos a cargo de la doctora Patricia Elizabeth López, jefa del Departamento de Inglés, para que haga la presentación de nuestro invitado del día de hoy. Doctora Pati, adelante.
Sí, muchas gracias, Bere. Muchísimas gracias a todos nuestros asistentes del día de hoy a esta interesante plática. Quiero agradecer de manera muy especial a las doctoras Laura Romero, Pati Díaz y Berenice Cuevas el apoyo y entusiasmo para que esta plática se lleve a cabo. Y por supuesto, a la doctora Lauri Martínez Chavarría, nuestra ponente del día de hoy, pues doble, triple agradecimiento por dedicarnos su tiempo, su entusiasmo y sobre todo su conocimiento, de su conocimiento tanto veterinario como bilingüe para compartirlo hoy con todos nosotros. En especial bienvenida a todos nuestros alumnos, profesores y público en general, pero en particular a los alumnos. Recuerden que aquí con esta plática estamos promoviendo, estimulando pues, dos aspectos muy importantes para todos ustedes en la formación de todos ustedes. Por supuesto, el conocimiento veterinario, pero además el aprendizaje de la lengua y que ustedes vivan el inglés en una manera ya real, acercándose a la medicina veterinaria, que ustedes vean a sus maestros de la carrera hablando en inglés, que ellos nos hablen que a través de esto nosotros conozcamos su experiencia, su acercamiento con la lengua y que ustedes vean hasta dónde pueden llegar, lo que ustedes pueden alcanzar con el conocimiento de esta. Bueno, antes de cederle la palabra a la doctora Lauri, dos indicaciones, por favor. Al final de la plática, al final de la ponencia, todas sus dudas, comentarios, preguntas que ustedes quieran hacerle a la doctora, lo pueden hacer en inglés o en español. ¿Sí? No es necesario que se hable todo en inglés. Si ustedes quieren resolver alguna duda, hacer algún comentario, expresar alguna opinión, lo pueden hacer en español. No hay ningún problema con eso. Otra cosa, por favor, los alumnos de comprensión de lectura, de la materia de comprensión de lectura que estén aquí presentes, les vamos a pedir que en el chat apunten su número de programa de la sala de inglés y su nombre completo, ¿sí? Recuerden que la asistencia a esta plática se toma en cuenta como horas de estudio, ¿ok? Entonces, sin más preámbulos, cedo el micrófono a la doctora Lauri.
Luari, mi nombre es Luari, ¿eh? ¿Sí me escuchan?
Se escucha perfectamente, doctora.
Voy a quitar los controles de aquí porque... Okay, well, good morning, everyone. Thank you for joining. I'm Dr. Larry Martinez, and well, I'm from UNAM, and today we'll explore how bacteria, these tiny warriors, as I call my talk, manage to cause disease. So first, a little bit about me. I work here in the Department of Pathology at the UNAM, and I did my graduate students both master and PhD in the Biotechnology Institute of the UNAM, and I have been working with pathogenic bacteria since ever. My postdoc was in the Washington State University in USA, and I worked first with Brucella when I was undergrad, and then my graduate students did it with working with Salmonella. Then my postdoc was working and researching on Yersinia pestis, the causative agent of the bubonic plague, and Today I work with Klebsiella pneumoniae and other pathogenic bacteria. So don't do the math, but I have been working around 20 years so far with pathogenic bacteria, and I'm interested in studying the molecular mechanisms that regulate bacterial virulence to understand their relationship with the host. Um, Here's the roadmap. First, a short introduction to basic concepts about pathogenic bacteria. Then, whether they need to be pathogenic. We will also explore virulence factors and why is it important to study bacterial pathogenicity. At the end, we will talk a little bit about the future directions and the closure. So before we dive into bacterial pathogenicity, let's explore our preconceptions. So let's start with a quick poll. I didn't know if I was going to be able to handle the controls or the Zoom surveys, so I put the link here. You can scan, please, this QR, and you
Doctora,
will see
¿quiere que
the
la haga,
question.
perdón, doctora, ¿le quiere que la haga administradora para que pueda mover y pueda trabajar?
No, it's okay. It's only this one, and I think it's easy to scan the QR, and the questions are gonna appear here, so we all can see the answers. So when you hear the word bacteria, what comes to your mind first? Just put one word, one or two words. Microorganism, disease, unicellular, right? Agent, gram, stain, obviously. Okay, that's all. Oh. None of you thought about something good? Microbiota. Yeah. Benefits for the host. Okay. Pathogenic. Biotechnology. Okay, so some of you think of disease and illness, and that's exactly what we'll talk about today. The few that cause trouble among the many that don't. What makes some bacteria truly pathogenic? So bacteria are literally everywhere. Soil, oceans, our beds, even our phones. They range from 0.5 to 5 micrometers. Imagine millions of these tiny microorganisms feeding on a pinhead. And only about 1 to 3% are pathogenic to humans or animals. The rest help us for digestion. They produce vitamins. They protect against harmful pathogens. And so yes, most bacteria are our roomies, not our enemies. And a funny fact that in terms of biomass, they are 90% of our cells. So who is the roomie now? No, it looks like we are the roomies. Mm. Okay, two words that we use all the time are pathogenicity and virulence. Pathogenicity is the ability to cause disease. It's a yes or no question. Virulence is about degree, how severe the disease can get. So a bacterium can be pathogenic, but mildly virulent or extremely virulent, like anthrax, for example. Therefore, pathogenicity is going to be a qualitative measure, and virulence is a quantitative measure. How severe is the disease? And both characteristics can change through genetic mutations, to acquisitions of different kind of genes, and uh, because of environmental pressures. So this is a dynamic process. How pathogenic is or how virulent is the bacteria. And in general terms, a pathogenic bacteria is gonna be the one that has the ability to cause disease in its host under certain conditions. This is also very important to remind. Do you think bacteria are born pathogenic or become pathogenic? You can use the chat to write your answer. I can see the chat. I don't know why. Okay. Become. It's difficult for me to see the chat while I'm looking at the screen. But most of you, okay, things like it's letter B. Sophia is not sure. Okay. Okay, exactly. Like most of you think, most become pathogenic. They acquire genes from other bacteria through horizontal gene transfer. This drawing shows a genome of a non-pathogenic bacteria. So for the bacteria to become pathogenic, it has to acquire genes by horizontal gene transfer. These genes come from the environment or from another bacteria. And when the bacteria acquires these genes, now, now it becomes pathogenic bacterium. But this is not enough. It's not enough to acquire these genes. And with this, I can tell you that bacteria, like it says here, don't start evolve. The non-pathogenic bacteria has to acquire virulent genes through horizontal gene transfer. And as I said, it's not enough. The bacterium has to learn when it should express them. That is, when it should turn them on, or if it has to keep them off. Because many of the genes they acquire can have detrimental effects also. But these genes aren't always on. So the bacteria has to make a smart regulations to decide when it's correct to turn on these genes, or to decide if these genes are going to be necessary for something. Okay. So they only express these genes in the correct location and at the appropriate time. Some people talk about the intelligence of the bacteria because they have to know bacteria are energy smart. They turn virulence on only when it's worth the effort. And sometimes these acquired genes contain the manual, that is, they contain the proteins that turn them on or off. But other times, the bacterium has to adapt genes that it has already to control the expression of the acquired ones. And so there can also be communication between different genes that it acquire, making the regulation of gene expression a very complex but interesting phenomenon. What tells bacteria it's time to express their genes? Because I already told you that it has to decide when is the perfect time. when and where? Well, temperature, pH, nutrients, oxygen, all are gonna be acting like signals. When they feel body temperature or low iron, they realize they are inside a host. And in that moment, they activate pathogenic programs. And again, they only express the correct location and at the appropriate time. Here at the right, you can see a diagram showing the colonization sites according to the nutrient gradient. For example, glucose is here. The gradient from red to blue indicates the concentration of glucose or nitrogen. Pink, high glucose here and in red, and low glucose in blue. And on the contrary, nitrogen is the high concentration is in blue, and the lower or the lowest is in red. And you can see that different conditions only for with the nitrogen and glucose change the bacteria that can live that live there. Helicobacter pylori is going to be here, whereas Campylobacter jejuni is going to be here. And this is depending on the amounts of glucose or nitrogen that they found. So you can see that how all the environmental triggers the appearance or the survival of different kind of bacteria. And pathogenicity is a three-part equation. Here it is, the pathogenicity equation that includes the pathogen factors on one side. This is the virulent genes that we are going to see, all the genes that cause for toxin, adenosine, bacillin, all the factors that they need to establish a successful infection. But on the other part is the host. How vulnerable is this host? Vulnerable animals due to age, immunosuppression, stress, are gonna favor the infection. So this is depending on the immune system of the host. And the third part of the equation is the environmental conditions. All the factors that we already saw, temperature, pH, oxygen levels, etc. All these are going to influence the survival and the establishment of the infection. Now let's see how infection actually unfolds step by step. Here is the general roadmap, and the parts or the steps that the bacteria has to go through to establish a successful infection are obviously the initial part, where bacteria must find ways to enter the body through cuts, nose, mouth, the digestive system, and they have to overcome physical barriers like mucus, cilia, tight junctions to gain access to the deeper tissues. Then the adhesion, detachment, and colonization. In this part, the bacteria must first stick to host surfaces. They use different proteins to attach to host cells and then establish the initial colonization. Then it has to penetrate host defenses and invade cells. Obviously, they also have to avoid the immune system and the destruction. Then damage and disease. In this part, bacteria can produce toxins and other harmful factors that directly damage host tissues or trigger excessive immune response, leading to clinical signs and pathological changes. So in this part is when we know the effect of the bacteria. And then they have to persist and spread. And in this part, we're gonna see the formation of biofilms and many other survival strategies that enable long-term colonization and transmission to the new hosts. Some of the factors that I will tell you participate in more than one of these steps, obviously, as you will deduce throughout the presentation. So we'll walk through this. Before a bacterium can attach or invade, it needs to get there, right? So motility is going to be very important to get there, to find their preferred niche. And for this, the bacterial flagella are so important because the motility is going to be often mediated by the flagellum. And bacterial flagella are classified into 4 types, and this is based on their arrangement. I mean, it's depending on whether it is one or several flagella and whether they are on one side or both of the bacteria. So you can see that there are bacteria that are monotrichous. This means a single flagellum at one end. Lophotrichus, a bunch of flagella at one or both ends. Amphitrichus, when they have a single flagellum at both ends. And the Amphilophotricus, they have bunch of flagella on both sides. And Peritrichus, the flagella are distributed over the entire cell. Well, there is another category, the Trichus, that describes bacteria that have no flagella at all. And with this kind of flagella, they can move in different ways. So at the right, you can see some of the kind of movements that they can have. So it's the normal one, let's say the swimming, but there is also the swarming when many bacteria move together. The twitching is like when they attach to a solid surface and then retract. So the bacteria is pulled forward. The gliding, the gliding is when the bacteria secrete a slimy substance, and that slimy substance helps them glide smoothly over surfaces. And the sliding causes most of them because of the expansion of the bacterial population. So as the bacteria divide, the new ones push against each other. So this is not a real movement, let's say, but it's product of the expansion of the bacteria. Their speed is impressive for something microscopic. And to illustrate this, let's take a quick look at the bacterial race. Yes, this is a real competition under the microscope. And this is the paper where I saw this is called the Microbial Olympics. And this is a race of many kind of bacteria swimming freestyle. The distance, 100 micrometers. And I put, for you to know each bacteria, I put here the origin. And there are some bacteria from Japan, some other from USA, this one from Australia, and this one from Belgium. And on the right, you can see the type of flagella that they have. So, which do you think is going to win? Write the number in the chat, and we can see it now. You can just put the number of your favorite. One, 2, 7. Most of you go for the 7. Interesting. Do you know this bacteria, the number 7? Or just because the 7 is your favorite number? 3, Vibrio. Number 7 is Rhodospirillum rubrum. Do you know it? Or why you choose the 7? It's interesting. 4, Vibrio, Acinolaticus. And you can also see if they are a puller or a pusher. This is funny, but that means the flagellum is gonna pull the bacteria. That is, the flagellum, pulls the cell, the body of the bacterial body along behind it, or a pusher when the flagellum pushes the bacteria along in front of it. Okay, so let's see who wins for freestyle swimming. Yes. Okay. So let's see who's the winner. Did you see who was the winner? 7 because it has two forces at once, Puller and Pusher. What did you think about the 7, Daniel? So who voted for the number 3? Well, now it's lower. I think the one that is the winner is number 6, right? Rhodobacter. Do you agree? It's difficult for me to see the chat at the same time that I'm looking at the presentation. Right, Dr. Lara, but it's 7 would be good for other things. Yeah, and if you are making fun of this poor guy, the number 3, that looks like it's high or dropped or lost, don't do that, because that is medically important. It causes otitis and wound infection, and it produces a potent neurotoxin that you can acquire when you go to the beach, because it's a marine bacterium. The other one, the winner, which is I think Rhodobacterus pharyngitis, is a free living photosynthetic bacterium, and this has been used widely as a model for the study of photosynthesis. Okay, let's move on. The next, now the first step is the adhesion and the colonization. The stick and stay step. Once they arrive, bacteria must attach and hold on. For this, they use pili, adhesins, and fibrils. Those are like tiny hooks and glue-like proteins to stick to the host cells. And this step is essential because if they can't attach, they can be flushed away by mucus or the peristalsis or any body fluid. The adhesins can recognize and bind to specific receptors on the host cell, and this specificity often determines tissue tropism and, importantly, the host range. So this step of the kind of adhesion that a bacteria has explains why a bacteria can infect one host, but it can't infect another host. For example, this explains why Salmonella pullorum affects poultry, but Salmonella cholera suis affects only pigs. And it has been said that this specificity is because of the kind of adhesins they have. Well, the colonization process has to is necessary to overcome all the natural barriers that I already mentioned, and they have to resist all the kinds that the mechanisms that the host has to clearance the bacteria. If they can colonize successfully, then they are going to be able to multiply and establish the successful infection. Well, from here on out, in each virulence factor, I will write in red only some examples of those bacteria that use this factor. And the first one is the E. coli, all the group that are known as attaching and effacing E. coli, or E. coli, if you want. The correct could be E. coli because it's Latin, but we are used to mention E. coli, right? So all the pathogens that are among the attaching and effacing group of bacteria are precisely known as attaching and effacing because of the characteristic lesion that they produce. One of these is effect, the enteropathogenic E. coli. And this kind of E. coli attach to the microvillar border in the intestine, and they form a characteristic lesion that resembles cups and pedestals. That is the third one. So once a pig found the correct environmental conditions, it starts doing the initial adherence. This adherence can be done because a specific protein, which is known as BFP. But once the bacteria is firmly attached to the intestine, they inject proteins inside the cytoplasm of the host, and these proteins are gonna induce that the cytoskeletal protrudes the membrane of the host cell, doing this lesion that is again called cup or pedestal because it's like a cup or pedestal. And if you see the real image of this characteristic lesion, look like this. So look, the bacteria is sit on the top of this cup or this pedestal, and in this other photograph down at the left, you can see the microvillar border on the of the colonic epithelium, the luminal surface, and the arrows are indicating all that line of E. coli attaching and facing this intestine. Ultrastructurally, E. coli attached to the microvillar border with the formation also of this characteristic cup and pedals. And for all of you that are in anatomopathology, these images can be found in this conference of the JPC. In urinary tract infections, there is another E. coli. Oh, sorry, my battery is getting low. Let me, I didn't realize that. Sorry, give me a minute. Okay. In urinary tract infections, there is another E. coli. This is the UPEC, uropathogenic E. coli. And this uses a special pili called P. pili to grab onto cells in the bladder wall. And this pili acts like a Velcro because it resists the flow of the urine, and that lets the bacteria cling upward. And that is why these infections, these urinary infections, are so hard to treat when this bacteria is involved in this infection. Well, after attachment, some bacteria literally break in, so they produce enzymes like hyaluronidase, collagenase, lecithinase that dissolve the tissue barriers. Others trick the host cells into swallowing them through endocytosis. It's both chemistry and manipulation, let's say. Well, the final goal is to get into the cell. Again, some are gonna induce the endocytosis, and some are gonna break down all the connective tissue in the on the surface of the tissue to get inside the tissue. And talking about manipulation, here's Salmonella doing just that. Salmonella is a very good model because it also has a syringe that injects effector proteins, and these proteins reorganize host cell actin, creating membrane ruffles that you can see here in the A and C. These ruffles engulf the bacterium and facilitate cellular invasion. And in this case, the same as EPEC or RUPEC or EPEC, they use a type 3 secretion system. This secretion system are called injectisome because they look like a syringe, and using this syringe, the bacteria can inject these proteins that cause the host membrane to ruffle, and engulf the bacteria. Here at the bottom and on the right, you can see the Salmonella invading the intestine, but invading because first they induce these membrane ruffles to engulf them and then internalize them. And this secretion system that I am talking about are like these syringes, as I told you. They are called injectisomes because of the form that they have, like a syringe. And these are systems to inject invasion proteins directly into the host cells. So in this diagram, you can see that at the bottom is the bacterial cytoplasm, and on the top could be the host cell. So when the bacteria again finds or detects that there is the correct place, they induce the expression of these secretion systems, and then when the syringe forms, it protrudes and injects the specific proteins since from the cytoplasm of the bacteria through the syringe until they reach the host cell. In this short video, you're gonna see the exact moment when Salmonella expresses all around their membrane the touch strip secretion system. On the right is the image put in both, and the injectisomes are indicated with these little blue arrows. And on letter B, you can see the structure of the type 3 secretion system. So put attention on the video on the left, because it's too short, but you're gonna see here is the Salmonella minicell, and all around the Salmonella are gonna appear this secretion system. You can see how the membrane protrudes, and then the injectisomes are expressed. Obviously, the bacteria is not gonna express only one secretion system. As you can see, all around the membrane, these systems are all around the membrane. And they let the bacteria inject the specific proteins. In this case, the proteins that are injected are gonna be useful to all the rearrangements of the cytoskeleton that induce the engulfment of the bacteria. Well, once inside, the bacteria must survive in attacks. One strategy is the capsule. You know that the capsule is a sugar coating that hides them from immune cells, and it prevents the bacterial recognition, and it enables. I told you that some of these virulence factors act on many steps of the infection process. So the capsule it's helpful to both to the attachment to the adherence and also for the evasion strategies. So thanks to this capsule, some bacteria can adhere to medical devices and the tissues. And Klebsiella pneumoniae, it's a good example of capsulated bacteria. This bacteria makes a thick, sticky capsule that prevents phagocytosis and helps it persist in hospital and clinics. In fact, Klebsiella pneumoniae is one of the most, not dangerous, but most of the important bacteria, nosocomial infections caused by bacteria. And you can see or you can test how powerful is the Klebsiella in its capsule, because there is a test, the string test. It's a simple test for checking the hypermucoviscosity of Klebsiella. You can touch a colony with a loop in the laboratory, and the string test involves stretching a bacterial colony with a loop on the agar plate, and a positive result is a viscous string greater than 5 millimeters. And you can see, all the bacteria that have capsule produce these mucus colonies on agar. Other bacteria change their surface appearance constantly, like criminals switching outfits. It's the same, and they do antigenic variation. They change all the surface proteins regularly to as I say to to disappear momentaneously of the host. This phase variation switches between different phenotypes because each time that the bacteria changes the proteins on the surface, the immune system uh, is not able to recognize them anymore. And the best example of this is Anaplasma marginale. This bacteria varies its surface proteins so the immune system can keep up. And there are many groups working on this bacteria. They produce bacterial peaks, characterized by expression of unique variants. Here you can see this is the genome of the bacteria. And in this particular section, they have many options to recombine. So this is also helpful, all the colors. And here you can see that when bacteria do recombination, it can take, I don't know, the blue with the red, and then the blue with the red and the yellow, and it's another variant. Or maybe the yellow with the blue with the black and the red, and each time it recombines in a different way all these proteins, it's gonna be another kind of bacteria, let's say. Okay, this is gonna be the surface of this protein in this bacteria, and each protein is different, as you can see. So the all the bacterial peaks are gonna induce a different response of the host, and this happens again and again. These are the bacteria in the blood to see the bacterial peaks, and these mosaics generated are gonna induce different peaks. And this happens again and again. And that is why this is so important, because this is inside a cow. And sometimes these cows show clinical signs and then nothing. And in this time is because the apparently nothing happens is because the immune system finally recognized that variant. But then the bacteria produce another different variant, and then it's another bacterial peak. And this can be for many months. Okay, talking about the damage to the host, we have been talking about many virulence factors that maybe are the purpose of the bacteria is not to damage the host. The bacteria is only trying to survive. But when we talk about enzymes and bacterial toxin, these can be considered a real virulence factors because they really damage the host. And the best example is the toxins. There are endotoxins, which are the lipopolysaccharide of the bacteria, which per se is gonna induce a massive inflammatory response. But there are other toxins which are called exotoxin, and these are the real secreted weapons because they are actively secreted by the bacteria to cause specific cellular damage at distant sites from the infection. And toxins um, are produced by many kind of bacteria. The well-known Shiga toxin from the E. coli again that causes hemorrhagic colitis and kidney damage. The other non-desirable toxin, the lethal toxin of Bacillus anthracis that causes anthrax and causes tissue necrosis. And this aflatoxin of Staphylococcus aureus that causes also hemolysis and cell death. But toxins come in flavors because there are cytotoxins, neurotoxins, and enterotoxins. Obviously, the cytotoxins destroy cells by making holes in cell membranes. The neurotoxins attacks the nervous system, causing paralysis. And the enterotoxin target intestines, obviously, causing severe diarrhea and dehydration. Clostridium botulinum is a good example of the neurotoxin. It blocks nerve signals and cause these huge effects. And here are the a chart that describes the toxin distribution. As you can see, most of them are exotoxins, and then the toxins and the little ones are the neurotoxins. Clostridium perfringens is a classical bacteria known by its production of toxins. All the principal weapons of this bacteria are the toxins. They are different kind of toxins, and all of them causes pores on the host cell, and that's why the cell dies. This is a image of a segment of the intestine, and note that loops inoculated with the toxins are severely hemorrhagic and distended with fluids. But when you remove the toxin of the bacteria, so now there are no significant growth abnormalities in these loops. Survive and spread. Spores is going to be one of these powerful virulence factors. The bacterial spore life cycle consists of two main stages, sporulation and germination. And this cycle allows certain bacteria to survive extreme environmental stress by forming a dormant structure called an endospore. And when conditions are favorable again, then the spore revives and returns to its active vegetative state. Some bacteria form spores or develop stress response systems that allow survival in harsh conditions. So, especially in harsh conditions, is the requirement for this bacteria to produce the spores. And this obviously enables transmission through environmental reservoirs and survival during antibiotic treatment. But if we are talking about persistence, the biofilm is going to be the star. Maybe you know what is a biofilm. These are called bacterial cities. And to survive long term, bacteria cooperate. So they form biofilms. These communities are enclosed in a slimy matrix, like a city. And inside, they are protected from antibiotics and immune cells. And bacteria also communicate. This is called quorum sensing. This quorum sensing is a system of cell-to-cell communication used by bacteria to sense their population density. And according to that density, they coordinate collective behavior. They do this by producing and releasing chemical signals molecules called autoinducers. And when the concentration of these molecules reaches a certain threshold, it triggers a synchronized change in gene expression for the entire bacterial population. And this allows bacteria to act like a multicellular organism, activating those processes such as virulence and obviously the formation of the biofilm. And why is important this production of biofilm? Because bacteria embedded in this matrix provide up to 1,000-fold increased antibiotic resistance and protect against immune system attacks. And obviously, this is gonna allow persistent infections. And here you can see how it is forming. They have to do an initial attachment, and again, biofilm is also an attachment virulence factor, but also a persistent or survival virulence factor. And first, a microcolony is formed where the bacteria multiply and start to produce these extracellular substances, this matrix, and in the final step, the biofilm matures. Here, the architecture is complex. They have nutrient channels, and the bacteria then can disperse can detach from the biofilm and disperse to colonize new sites and start over a new biofilm. In this little video, you're gonna see how the biofilm grows. This this group in they study different surfaces where the biofilm can be produced, and they test different concentrations, different agar concentration. This is 0.6%, but they prove a lot of concentration, and they are a group of uh, um, physics that is study how is the collision geometry of this biofilm. Here is the lateral view of the biofilm, and you're gonna see how thick, how thick can be. And they study this, trying to study the structure of biofilm, which structure is more fragile or more difficult to break down, with the objective to find mechanisms to contain the biofilm production. But it's so interesting. And in veterinary practice, biofilms are more common than we think. They form on teeth, catheters, implants, any moist surface can have a biofilm. And it has been reported that over 80% of dogs and cats develop dental biofilms by age 3. Once that slimy shield forms, antibiotics barely reach the bacteria. That's why it's so important to study the production of this microbiological series. And this links the talk with the antimicrobial resistance as a virulence factor. Installed biofilms bacteria, as I told you, is 1,000 times more resistant to drugs. And it's not only genes, it's architecture. The matrix slows drug diffusion and shelters dormant cells, so they are well protected from the antibiotics. And this is how the way that a bacteria become resistant. The at the beginning, it could be a lot of germs, but some are gonna be drug resistant, and those that are resistant are gonna be able to grow and take over, and then they are gonna be able to transfer that drug resistance to other ones. And bacterial pathogens are not only developing virulence mechanisms, but also they are acquiring resistance to antimicrobials, creating what we know like superbugs that are both highly pathogenic and difficult to treat. And these bacteria have evolved multiple mechanisms to survive antibiotic treatments, including enzymatic degradation, target modification, that is altering drug binding sites, expelling antimicrobials from bacterial cells, or forming these biofilms. This makes infection harder to treat and increasing the risk of disease spread, severe illness, and obviously the death. And antimicrobial resistance isn't just a human hospital issue, it's a One Health problem. Resistant bacteria move between animals, people, and the environment. If nothing changes, projections show that 10 million deaths per year are going to be expected on 2050. So, yes, some researchers have been have said that the next pandemic could be microscopic and slow. Uh, this is a video. I'm gonna put it on silence, that made at Harvard to show the population how the importance of the antimicrobial resistance. Here you can see a huge petri dish with agar, and they put on each line different concentration of the antibiotic, starting from nothing at 0, then 1 time, 10 times, 100 times, and 1,000 more antibiotic. And you're gonna see that the growth of the bacteria on both sides And at the beginning, the bacteria are gonna grow normally because there is no antibiotic. And then is a pause. Every time they reach a limit, a new line, they do a pause, and then one of the bacteria become mutant and start growing. And then you can imagine that these new resistant bacteria share the genes or the information that it has, pass through the other bacteria. Then there is a pause again each time they reach a line. And every time that there is a pause, the bacteria is adapting to the new condition, gaining or mutating, and then uh, becoming resistant. And at the end, you can see that even at 1,000 um, more concentration of the antibiotic, there are some bacteria that grows. And in this part, they are showing us each of these points indicates a mutant bacteria that now is resistant and able to pass information to the other ones. Impressive, no? This is a selection pressure and is favoring resistant bacteria. And but bacteria teach each other tricks, and resistance spreads like gossip, as you already saw. And in 2017, the World Health Organization published a list of priority pathogens to promote their research and thus develop new treatments to combat them. The list consists of 12 bacteria, but is headed by a group of 6 pathogens responsible for the majority of nosocomial infection worldwide. These are the escape pathogens, and these are the rock stars of resistance. These are Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and different species of Enterobacter. And since 28, the acronym ESCAPE has been implemented because they literally escape every treatment. And some infect pets, livestock, and humans. That is a proof that boundaries don't exist for microbes. And look at this. This is the percentage of resistance uh, through the time. So look in 2018 and now is the trend is constantly increasing. So well, finally, I would like to read your comments, and if you have any thoughts about why is it important to study bacterial pathogenicity after all this that I have been talking about. I can see the chat. Any thought or comment? So now that we have reviewed several of the virulence factors, I would like to add to this because it is important to study all this, in my opinion, to better understand the behavior of a pathogen and therefore know what we can expect when we identify it as a causal agent of a given infection. And to explore this, I'm gonna let's review these two cases, for example. Imagine that you have a dog. It's a canine pyoderma. 5-year-old golden retriever presents with recurrent skin infections. And Staphylococcus intermedius has been isolated. And these bacteria show biofilm formation, beta-lactamase production, and surface protein adhesions. What can you expect on this infection? Any thoughts? According to what I have been talking about, any thoughts? It will be a difficult infection to treat. Yeah, that is the main conclusion and the final conclusion, right? So now I hope that you understand that in this case, biofilm is going to be protecting bacteria from topical treatments, that while adhesions will enable recolonization, and that the beta-lactamase that was identified will confer antibiotic resistance. And yes, that will be a challenging treatment. The second case, equine respiratory disease. A racehorse develops severe pneumonia after transport stress. In this case, Streptococcus equi was identified, and it has M protein, hyaluronidase, and streptomycin on it. What can you expect? In this case, M protein will help the bacteria to resist phagocytosis, hyaluronidase to spread through tissues, and the streptomycin to damage host cells. So in this case, the combination of stress-induced immunosuppression and bacterial virulence factors results in a rapid disease progression, requiring immediate aggressive treatment and supportive care. And both are real cases. Which animal gets sick more? Why is this important? What do you think? You know which animals get sick most? It's a funny fact. Think about pigs, dogs, cats, cows. Which one do you young animals? And talking about the species, young animals is correct, and it's gonna be on any species. And talking about the species without the thinking about the age, well, I found that the poultry are the ones that are the top of the list. You know why? Because birds are in crowded conditions. Crowded housing makes them easy targets. The hopeful side that good hygiene can prevent up to 80% of these outbreaks. Well, the field is exploding with discoveries, new host pathogens, insights, smart vaccine, faster diagnostics, and understanding virulence at the molecular level helps design therapies that disarm bacteria without killing them, reducing resistance pressure. So by comprehending how bacteria cause disease, we can develop smarter strategies to prevent infections, choose appropriate treatments, and protect both animal and human health. And this is where our role as veterinarians is crucial in this global effort. Here is a chart showing the number of publications since 2019 to 2024. And as you can see, the field is increasing. 2,000% research growth rate. So the pathogenicity research is interesting. And here are finally some fascinating disease statistics. Look, you can see the economic impact. There's already has been said 1 to 3% are only the pathogenic bacteria species, but some numbers to put things in perspective, right? Behind those bacteria are real economic and welfare costs. Bacterial disease is also threatening wildlife, from koalas to coral reefs. Chlamydia affects 50% of all the koalas in Australia, and microbes really rule every ecosystem. I would like to finish with this quote of Stephen Hawking. The greatest enemy of knowledge is not ignorance, but the illusion of knowledge. And I think that in the rapidly evolving field of bacterial pathogenicity, maintaining intellectual humility and curiosity is essential, because new discoveries continuously reshape our understanding of host-pathogen interactions. And this reminds us constantly that learning is a lifelong journey in veterinary medicine. So thanks for your attention. I hope this talk gave you a clearer and maybe more respectful view of bacteria. They're not evil, they're just extraordinarily good at surviving. So feel free to email me if you'd like to continue the discussion.
Gracias, doctora Lori. Si alguien tiene preguntas, como les comentó la maestra Patricia, podrían hacerlas ya sea en el chat o bien abriendo su micrófono, levantando la mano para mantener el orden, por favor. A ver, tenemos una. Luis, adelante.
Thank you, Doctor, for your speech. I would like to ask you a question about Salmonella vaccine in pigs. Maybe this could sound rhetorical, but I would like to know what's the difference or the advantage and disadvantage of vaccines given via drinking water versus injections.
Which is advantage of each one?
Yeah, advantage and disadvantage of Salmonella vaccine given via drinking water versus injectable vaccines.
Well, I really don't know the exact response because I'm not sure what is inside each vaccine or each kind of vaccine. I don't know with the serotype is in the vaccine that is the one that producers are using on pigs. But if you only ask me about the 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 way, the difference between the oral or the injected vaccine, I could think that as many other treatments, oral administration has to be very nice protective, no, because as you saw, the bacteria has to contend with a lot of environmental factors during its transport through the mouth and along all the digestive system. But I'm not familiar with the components of each vaccine. Sorry, I only can think about the resistance administrating the bacteria by oral administration. But I'm not familiar with the serotypes or the other components of the vaccine.
Thank you, anyway.
Doctora Laura, adelante.
Oh, thank you. There is a comment in the chat from Victor Daniel who says, I have always had the problem with many colleagues about the way antibiotics are used, and I've always seen a lot of disbelief from doctors. They prefer to continue using medicines indiscriminately, even as prevention, rather than risk having complaints from people. In my opinion, this should be information that should be handled to the entire population. Agree.
The worst thing that many agencies have shared the information, no, and the WHO, the World Health Organization, is tired of sharing all kind of information to the doctors, to the laboratories, to the schools. But as you said, Daniel, they don't even care, no, because sometimes they prefer to prevent an infection because they are afraid that the animals can be sick, can get a disease instead of prevent another thing that could be worse. Because these statistics for the future are real and are based on many data along these years, but people don't believe doesn't believe that this is gonna happen. No, this pandemic that is gonna be, and it's gonna be awful. There are many researchers that have been told that in 2050 or 2030, we all are gonna die because of a diarrhea that is not gonna be treated by any antibiotic.
Yeah, it is unbelievable that we're supposed to be or to have more information because we are health professionals, and it's just, It's crazy. There is another comment from Carla Mollinedo in the chat. She says, sometimes the route of vaccine administration enhances the production of IgA instead of IgG, which could be an advantage.
Yeah, that's right. Okay.
And there is another one from Elizabeth. Besides disinfection or hygiene methods, are there any therapies targeted toward biofilm?
Any of the therapies is effective. Any. That way the worries about the biofilm. And every time in human hospitals, any time that they discover a biofilm is formed, is produced in any medical devices, they prefer to remove the medical device because it's easier to remove and to buy and to remove the old advice and to put another new one, then treat the infection. And maybe you have heard, even when your family, when you have a family inside the hospital, the doctor said, if your familiar is getting better, take it. Take it because it's worse to be here inside the hospital because the risk of acquiring a nosocomial infection is really high. I have many friends that work in a human hospital, and they also said they always said no. The most the higher infections are nosocomial, and most of the patients die because of secondary nosocomial infection rather than the original infection.
And this
No,
is,
and no, and that's the reason that there are a lot of people studying how to break down the biofilm. But the thing about it, the biofilm is not only the bacteria; it's a matrix. There are a lot of sugar, and sometimes the biofilm is not composed only for bacteria. The biofilm also has fungi, viruses, protozoals. They are a complex really series, and they protect. And sometimes the antibiotic can kill the bacteria that are on the surface. But this bacteria died in to protect the other ones, no? And that's the other thing about the bacteria, that they help each other, and they are able to sacrifice itself just for the rest of the bacteria can live. And this is what happens. So that's the reason that nowadays there is no an effective therapy against the biofilm.
wow. Oh, another thing to worry about. How do you think, there's another question. How do you know when the bacteria have developed a biofilm, like in the intestine?
How do you know? Because you can see the biofilm because the population of the bacteria is huge in this biofilm. But unfortunately, the first thought that comes to think, maybe it's a biofilm is sadly when the treatment is not working.
It isn't working, yeah.
Yeah, yeah.
That's well, there are no more questions in the chat. Oh, actually, there's another one. Are there any cases in which a bacteria that usually infects only a certain animal species infects a different one?
Yes, yeah, and that's very common because sometimes, as I told you, there are the horizontal gene transfer. And thanks for your question, because many times we are used to know that this bacteria produces this disease and in this species, no, and we are like mm, very strict on thinking that, but please remember that all the bacteria can transfer genes. And the bacteria that you are used to know that affects only certain type of animals now is infecting another ones, or maybe the same animal but in a different presentation, because now it's in the stomach or now it's in the causing pneumonia, and that happens. And that happens every time that you see that, and there is a clinical sign that doesn't check with what you know about that disease. No, think that the bacteria evolve transfer genes. And yes, there are many, not many, not a lot, but there are cases, there are frequent cases where the bacteria now is acting different, and now it produces it induces in the host different clinical signs.
Okay, well, we have comments about the how great your talk was. Thanks a lot, Larry. It was really very, very interesting and fun, and yeah, it was amazing. Thanks a lot for sharing.
No, thank you for inviting me.
No, thank you, thank you. It was really, really good, and you have lots of comments, like very good comments about your talk. Thank you.
Por último, tenemos a Luis, que yo creo otra vez, o se te levantó la mano sin querer, Luis.
No. I want to make another question, because the last thing you said about these new diseases that are affecting animals to others made me question me about if this could lead to a new kind of new zoonosis.
Yes, in some cases that could happen. There are some cases when you find a bacteria that is in human, for example. As I told you, I have some friends working in human hospitals, and sometimes they have found bacteria causing the disease in that person, and looking for tracing the bacteria. Sometimes they found that bacteria was in their dog or their cat or their bird, and in that's why we work together, because once they found those kind of infections in humans, talk to me and we work together, and we have found that sometimes, but sometimes those bacteria come from the animal, from the pet of that person that now is infecting the human. Obviously, most of these cases are showing in persons that are immunocompromised, no? But it can, all these mechanisms can generate new zoonosis.
Yeah, that sounds interesting, because in my working place there are a lot of pigs, and well, we know about the zoonosis that we are sharing, but this made me think about the chance that we can get a new disease from the animals. Well, I was gonna say boars, because I work with boars, but maybe they can get a disease from us if we are not having this clean action that we have to have in order to in order to keep all the health in the on the herd. I'm talking about shower in, shower out, washing hands, disinfecting, a lot of the action that we
all those measures
have to make.
exist because of that. It's totally possible. It's not impossible. All the existence of all those measures, the reason of all those measures is precisely the risk that you have to carry an infection to your animals, and vice versa, because sometimes it's in both ways. No, you can get an infection from animals, but you can also spread one of your bacteria or your infections to your animals or different animals.
Yeah, because the other thing that I agree with you about, we are used to see a disease with this kind of presentation, but sometimes it doesn't correspond that what we have been reading on the books or on class or whatever. And this is something that it's giving me the creeps, because I'm in direct touch with those animals, And you know, I have seen a lot of signs of disease in them, since scours, coughing, a lot of infections, you know. So, and this is making me think about this chance to maybe get a new disease that maybe we don't know yet, or maybe we don't know the source of this disease, because sometimes we can think that it's, oh no, it's a kind of the environment, it's an allergic reaction, and sometimes and maybe this could be the chance that is another possibility to get this new illness sensation.
Yeah, and I'm glad now you think about it, because sometimes that could be possible, because we know how a disease behaves, no? But as you already mentioned, maybe this is not what I have read about this disease, no? But talking about bacterial diseases, my recommendation is that always find the causal agent, because sometimes some clinical signs or some histopathological lesions could orientate us. Yes, it's true, but until you don't have that the real causative agent is this one, you are not sure. Everything is possible. So my recommendation is that when you suspect about a bacterial disease, find the bacteria and be sure of what bacteria is the one that is involved in that case. And from that from
Why?
from that moment, you can study that bacteria and you can investigate them, and you can see which virulence factor that bacteria has.
You can treat it correctly.
Yeah, well,
Exactly, and most important, you can treat
I
it
know.
correctly. Mhm.
I know, but that can be a problem, because since this is a company, a big company, they have a strict protocols for treatment. So if we have this uh, kind of disease, you have to treat them this way. You can give them another one, but the one that they are telling you. That's one thing. The next thing is that every time that we have any sign of disease and we are taking samples and send them to the lab, they are searching for an specific pathogen. No, they are not seeking what is causing the problem. So if it's not positive for Salmonella, Brucella, another bacteria that is probably make an alert to the government or to them, in order to don't ship or move the animals from one side to the other. It's not, they are not worried about. I mean, if this is not a disease that can be a problem for the herd, they don't care. I mean, if they are searching for a specific pathogen, and
And it's good because at the
now
beginning
that
you're
you're
gonna you're
mentioned,
gonna have your first option. So it's not bad.
yeah, and
Obviously, the clinical sign are gonna guide you, no? So you have
uh-huh,
your main suspects, and it's okay to look for
yep,
for them first. Is a good way to thought about the solution, no? But once you detect that this is not working,
yeah,
or this treatment is difficult, this treatment is not working anymore, look for anything else.
yeah. Well, something that I forgot to mention is that I'm not living in Mexico. I live in the United States. So that here in this country, we are not bats. We are just workers, and it doesn't matter if you have a degree or whatever. Uh, that's why I was telling you that they want us to treat the boars with that specific treatment. So there's not a looking to the other side. No, it's a straight view. If you see that sign, give them this. If that is not resulting, there's a second option, but that's it. And sometimes they don't get better. That's a problem. And sometimes it's not the proper treatment because, well, I know for sure that it's not the right treatment, but at the end is what they want us to do.
Yeah, I understand. It's difficult, no? Because there are other
very
qualities
difficult.
there.
Absolutely.
Many factors to
Absolutely.
consider.
But, thank you. I don't. Yeah. Thanks so much for your time.
No, thank you.
I will let you go because I know it's late.
It's okay.
Bueno,
Thank you.
muchísimas gracias a todos los que participaron. Maestra Patti, si quiere despedir a nuestra ponente, la doctora Luari, adelante.
Bueno, ya se lo puse, se lo acabo de poner en el chat. Muchísimas gracias, doctora, por compartir, sobre todo con nuestros alumnos, su conocimiento, su experiencia, y pues esperamos que esto sea muy inspirador para ellos, para que sigan sus pasos.
No, muchísimas gracias a ustedes. Me parece que estas actividades son buenísimas para todos, para que unos practiquemos hablando, para que otros practiquemos oyendo. Las felicito por esta iniciativa, ¿ves? Porque es muy bueno. Mezclas todo, ¿no? El idioma y también, pues, conocer sobre algún tema. Gracias y